Hi everyone, welcome to another week of the Behavior, Evolution, and Culture Speaker Series. We meet every week in this room at this time, except for next week when there's a holiday and we won't be meeting. So please sign the sheet that's going around. That's how we keep track of how many people are attending. It should be here somewhere. Is it? Okay, it's going around. Great. And if you want to join the email list, I see new faces here, so just put your email on that list and you'll get added. And so next week we don't have back. The following week, Nandita Garud is speaking. She's from a new faculty member in the Department of Ecology and Evolutionary Biology. And this week I'm pleased to introduce Angela Garcia, who's a postdoc at Arizona State University, talking about the embodiment of stress.
Pardon, I'm going to stay right here because I have to keep forwarding the slides, but so sorry if that's a bit annoying. Alright, so I'm actually going to start by stepping back a second and telling you a little bit about where I came from and how I got to doing the research that I do, or rather kind of how I got honed in on the research questions I am, and also because where I came from really informs the way that I approach research, my research interests and evaluating these questions. So I spent a number of years before graduate school living and working in Detroit, Michigan. And I had a lot of different jobs. I worked in restaurants. I was a assault response advocate for the women's shelter. I taught ESL at an immigrant rights organization. I was a HIV tester and counselor. And through these experiences, I became very aware of something, that health is really unequally distributed. And who has health and who doesn't is quite different. And so this kind of led me to think about, well, one, what are the causal factors that lead to this kind of patterning of this unequal patterning of health? And also, why do we see this, right? So for instance, teaching ESL, the majority, or I should say exclusively, the people that came to that class were Hispanic immigrants. And what I realized was that almost all the individuals in those classes, either they themselves or had a very close family member or someone in their community that had diabetes. And very similarly with the Black community in Detroit. You see a lot of diabetes, you see a lot of obesity, you see a lot of high blood pressure. So a lot of these kinds of metabolic diseases. And actually even take a step back further, at the global level, metabolic diseases are the #1 killer, and increasingly so. So over the next 30 years, we're going to see a 30% increase in metabolic diseases. And the majority of that is coming from low and middle income countries. Now to again step back to the US, if you think about on the population level, 1 in 11 people over the age of 20 in the US have diabetes, which is, I hope that is a very startling number to you. But even worse than that, if you break it up by ethnicity, for Blacks and Hispanics, that number goes up to 1 in 5, right? And now from research, we tend to try to understand these or evaluate these issues with looking for kind of a smoking gun. Not because we want to, but it's really hard to incorporate all these different aspects, right? So we say things like, is it genes, right? So for instance, with the Hispanic immigrants, is there something like the thrifty genotype or thrifty phenotype hypothesis? Is that what's explaining this disproportionate number of Hispanics that have diabetes, right? Or is it something about the social environment? Is it poor sleep? Is it discrimination? And the problem with that is that when we start to kind of look at it in such a narrow pathway, we either see conflicting results or we can't understand the pathway through which we see these social agents leading to metabolic pathologies. So my research program really takes a kind of systems approach where it's incorporating a lot of elements from a lot of different systems. And so my primary goal is kind of looking at the way biological processes interact with each other, and then how social and ecological or environmental factors are influencing the way these systems interact, right? So for today's talk, I'm going to essentially be looking at the pathway through which social diversity can be linked to metabolic disease, and focusing in on the relationship or the influences that social diversity has on the processes of the stress response and the immune response system and how these systems interact to kind of fill in that pathway. Now so especially in our field or in bioanth and a lot of kind of related fields, it's also not new to us that there are links between the social world, stress, and the immune system, and metabolic disease, right? So we see things like
Depression is associated with 60% increase in risk for diabetes. Social isolation is linked to higher inflammation. Perceived social isolation is associated with upregulated glucocorticoid desensitization genes. And individuals from low SES backgrounds have higher levels of cortisol and inflammation. But why do we see these diverse aspects of social adversity converging on the similar types of diseases? And so what I'm going to really focus on today is looking at how these diverse social factors share common influences on certain systems in your body that regulate your metabolic homeostasis. So your stress response system, or your hypothalamic pituitary adrenal axis, and your immune system. And before we go a lot further, I'm going to just focus in for a second to kind of talk about how I'm going to be operationalizing your stress response system and your immune system in the context of this talk. Also, just as a heads up, if you guys have any questions, well, at least clarifying questions, you're very welcome to stop me. So for your stress response system, or your hypothalamic pituitary adrenal axis, activation of the HPA axis is one of the main ways that we think about physiological and psychological stressors influencing our biology. This is kind of one of the main neuroendocrine systems, and essentially, actually I'll show you this first. So you have your body, or I'm going to be actually focusing just on cortisol, which is a primary hormone of your HPA system, right? Cortisol follows a circadian pattern, and it has a diurnal profile, which means that shortly after waking, it spikes about 30 or 45 minutes after waking, and then it spends the rest of the day falling. But if you experience some sort of an acute psychosocial stressor, you have this spike, right? And so that system adjusts through a kind of a recalibration and release of a bunch of different kinds of hormones and catecholamines, in particular cortisol, that responds to that stressor, but then afterwards it falls back down pretty steadily. Now in your immune system, you have really two arms of your immune system, your innate immune arm and the adaptive immune arm. And these exist on a continuum. So you don't have to, I'm not going to belabor the details of this, but for the way I'm using or looking at innate immunity is through granulocytes, and for adaptive immunity it's lymphocytes. For innate immunity, you can think about this as the kind of fast response. It's actually pre-existing before you encounter a stressor, right? It's quick on the scene, it does its job, but it's kind of like throwing a bomb at a problem, where it resolves the problem, but it does cause a lot of collateral damage to the cells and tissues around that. On the other side, or the other arm, the adaptive immune system, this is kind of like the sniper. It takes a while to train. Your adaptive immune system doesn't really come online until maybe a couple days or a week after encountering a pathogen, but when it does respond, it's really precise and highly effective. And so it doesn't cause collateral damage, and it also does a lot of repair. What's quite interesting, and I think wildly underappreciated, not just in our field, but across a lot of fields except for really chronobiology, is that the majority of cells or components of your immune system also follow circadian rhythms. And they have a diurnal profile that's actually opposite or antiphasic to cortisol. So where cortisol is spiking in the morning, the majority of these immune cells are at their nadir, their lowest point, and they actually raise over the course of the day and peak during the evening. And so now, one of the issues with understanding how social factors relate to disease risk is that we tend to look at these systems in isolation, or kind of just cross-sectionally using just base levels or absolute levels, right? And the problem lies therein, right? So you have something like depression. Depression is associated with high cortisol. Depression is also associated with low cortisol. Depression is associated with immune function, but it's associated with suppressed immune function and overactive immune function. And so, you can kind of play these out, and you see all of these really confounding relationships. And what I'm going to argue today is, or in order to address this paradox or resolve this paradox, we need to start looking at the regulation between these systems, between the way your stress in your immune system, your HPA axis in your immune system communicate with each other and regulate each other, to understand kind of how these psychosocial factors are then related to downstream health effects. So to start with just HPA and immune signaling, this is a great paper by Dumble and colleagues, and it's looking at, so you can see there's a cascade of things from your HPA system that they upregulate some aspects of your immune system and they downregulate other aspects of your immune system. And then there's this kind of feedback loop where if one thing's upregulated, it might then send signals back to the HPA system, which then adjusts the release of those hormones, and it kind of, these systems keep each other in check. The way they're able to do that are through glucocorticoid receptors. And glucocorticoid receptors are these little receptors that are essentially in cytoplasm in cells, and they decode and pass information between the glucocorticoids to the nucleus in order to influence the expression of some of these immune-related genes. So you can think about this as just the kind of decoder and passer of information between these two systems, between the HPA axis and your immune system, okay? Now, I am also not the first person that realized that it's within these interactions lies a lot of the explanation of disease, or dysregulation of these systems, I should say. So this has been shown in a lot of different studies. But again, this primarily comes out of chronobiology. So you see delayed wound healing, impaired responses to vaccines, development and progression of cancer, and obesity, type 2 diabetes, and a lot of other metabolic syndromes. And even going just beyond these base relationships between how the immune system and the HPA axis talk to each other, they actually, the cortisol, actually in specific, regulates a lot of the diurnal components of your immune system. Now I'm going to get a little bit into that, a little bit more into that. So it both stimulates, it has stimulative and suppressive effects, but just the diurnal pattern of cortisol also regulates the diurnal pattern of a lot of your immune function. Now, kind of my approach to this is it's spanning and pulling across from a lot of different fields, right? I'm pulling from anthropology, biosocial anthropology, psychology, immunology, endocrinology. And I'm kind of uniting these things under an evolutionary framework in order to try to understand questions like why do most components of the HPA and immune systems follow diurnal patterns at all? And what can that tell us about when you see dysregulation in these patterns, why you might see dysregulation, and then how it might relate to downstream health effects. We can also ask questions like, why is the HPA axis responsive to immunological cues and also social cues? And how might this inform the patterning of metabolic diseases across populations and in contemporary environments? So I'm going to go a little bit deeper into theory just for a minute, and then I'm going to focus on two studies that I've done in Honduras looking at immigrant women on the island of Utila. All right. So first, why do most components of HPA and immune systems follow a diurnal pattern? This is a really awesome paper in Science by Kevin Mann and some colleagues, where they essentially posit a couple of really interesting things. One, if you think about over the course of human history and human evolutionary history, so like I was showing you on that past slide where you see most or a lot of aspects of immunity are kind of at their lowest point in the morning, right? And they raise and they peak at night when you're sleeping. Well, if you think about what is the utility of having up and down-regulated aspects of your immune function, if we are spending the early part of the day foraging, looking for shelter, you don't want to be bearing the cost of having a kind of overall upregulated immune system, right? Why not depress the aspects that aren't immediately useful, maybe keep some of them upregulated that are some of these innate responses, but otherwise just prioritize what's going to be most effective in the short term. So you downregulate a lot of aspects of immunity when your body is very active. And also you're capitalizing on the immunorepair, the aspects of your immune system that are peaking in the evening when you're resting. You're capitalizing on the fact that your body is not expending energy in other things, right? You can just use that energy to do those, well, first also upregulate the parts of immunity that are more costly, but also it allows them to do that repair without being kind of, without drawing energy to other sources. And then also, you decrease the potential for runaway immune responses. So the fact that you have all these diurnal patterns, and they're all slightly staggered from each other. Imagine if they were all kind of dialed up to 9 all the time, right? If you did, or when you did encounter a pathogen, the likelihood of your immune system just jumping and over-responding to that pathogen is a lot higher than if you have this staggering circadian pattern, or staggering diurnal pattern, where you can still reach the same peak when you do encounter that pathogen, but you're mitigating the risk of that over-responsiveness, and so mitigating the risk of things like sepsis or autoimmune disorders. Now, in addition to having that diurnal immunity, the HPA system also serves an additional function that further optimizes your immune responses. And it does this in that it regulates it regulates in response to a stressor, and then overall the diurnal pattern regulates the diurnal patterns of immune function. So it does help terminate an inflammatory response at like that acute level, right? If you encounter a pathogen and your immune system upregulates a lot of inflammation, it leads to this cascade of hormones that then kind of suppresses it and it turns that immune system off or dials it back down. But then again, when it's at its nadir, so over here in the evening, at its nadir, it's also releasing other catecholamines and things that are stimulating those immunorepair mechanisms. But in addition to having this kind of ecological function, the HPA axis also has a social function, where it's really highly sensitized to cues of social stress. And it actually responds quite similarly, where it is prioritizing these short-term costs, or not short-term costs, short-term benefits, upregulating things that are going to be beneficial in the short term, and downregulating things that are going to, you're not going to need them as much in the short term. So you see things like an increase in glucose production, but you also see a decrease in glycogen synthesis and insulin secretion, which essentially is keeping more glucose in your system and not allowing it to be stored effectively, which in the short term is good because it keeps that glucose ready and available for use in other ways. It also modulates the immune system similarly, where it's depressing or downregulating things like your adaptive immunity, which isn't going to be that important in these short-term social interactions, but upregulating things like granulocytes, which will be very useful. Now, this fight or flight response is likely optimized for short-term acute threats. And so if you think about the diurnal nature of these systems, it suggests that they evolved to respond to a certain amount of environmental variability, whether it's social, nutritional, ecological variability. And these systems are quite plastic, right? But what does this look like in these contemporary environments where you might have kind of a strained or chronic stressors that might be potentially novel, or just environments where you have a lot of too many calories or something, where you get these systems that have evolved under relative constraint, and you put them in a contemporary context where you have alleviated those restraints, but now perhaps they're not responding as adaptively as they were meant to. And so now I want to think about the effects of chronic stressors on these evolved systems. So chronic stressor, something like discrimination or depression, these are conditions that are characterized by having a lack of control or unpredictability, and they can lead to a sustained stress response. So while you might have the kind of normal stress responses, it peaks and then it falls, and it might peak again if you have an acute stressor. If you experience these chronic stressors, what it leads to is this kind of, it's called blunting of the system, where you lose the diurnal rhythm of cortisol, right? And it kind of flatlines. Knowing how your immune system knowing how your HPA axis regulates your immune system, if you have a chronic blunting of this system, what it can do is it can lead to a breakdown in the crosstalk between these systems. It leads to a desensitization of glucocorticoid receptors. And this leads to glucocorticoid resistance. This is essentially means that your immune system is no longer sensitive to the signaling by cortisol, right? And this is a major thing that precedes insulin resistance and diabetes. And this is what I'm going to, well, I'll get into this in a second, actually. All right, so to get into these studies, So I did a bunch of fieldwork on the island of Utila over the past 4 years, and kind of increasingly honing in on looking at the effects of stress and metabolic disease in women. Utila it is actually a Honduran island. I know that I'm talking about Honduran immigrants, and I'll get into that too. But so this is an island about 40 kilometers off the coast of Honduras. It's quite small, and the population is about 6,000 total, with 2,000 of these individuals being Honduran. And really, this is kind of a tale of two islands. So while that's a Honduran island, it actually didn't get given back to Honduras until the mid-1800s or so. And it is the native islanders, the Utilians, look like Uncle Roger over here. They are of British and American ancestry. They speak English. They speak Creole English. Their culture is heavily influenced by American culture. There's been a long history of import and export. And there's also a lot of Utilians have worked as merchant marines over the years. And so Utilians, also Utila is a heavily touristed island. They do a lot of dive tourism. Utilians tend to either own or operate dive shops or restaurants or bars, things that are kind of in support of the tourist industry. And they tend to live like this. This is kind of downtown Utila, where they live on the perimeter of the island by the ocean. Starting in about the 1990s, you got an not insurgence, that's a weird word to use, but you got an increase in waves of migration from the mainland Honduras to the island. And part of this is because there was just an economic downturn on the mainland Honduras. There was also some natural disasters that led to, and also the increase in tourism, there's a lot of push and pull factors that were bringing more Honduran immigrants to the island. And so this is kind of a typical area that Hondurans would live. And so they're not the owners and operators of dive shops. They are the more day laborers. They maybe clean houses or drive tuk-tuks or taxis, maybe work in the kitchen. So there's a really strong cultural and economic disparity between these two groups, where you see a lot of overt racism, discrimination, stigmatization of Hondurans. The majority of Hondurans live in an area called Camponado or Campo Lacho, which is the extension of Camponado. This is in the inland of the island that was built out over the swamp. And so they essentially packed in trash and coral rock, and then would put either sand or pour concrete over it to make the kind of paths, and then they build up houses on stilts over the swamp here. The issue here, though, is there's no wastewater management system. So all of the human refuse just goes through PVC pipes into the swamps under people's houses. And you have really high pathogen exposure, and we see this, that there's parasite infection is pretty high in the island in general, but the kind of base of it is in Camponado. You see a lot of parasite infections coming from there. So apart from this high pathogen exposure area, being from and living in Camponado is also really highly stigmatized. And so this is just a couple of kind of quotes to get at that. So this is from a dive instructor. This is the road to Camponado, which I pass every day, but I would never venture down. This is a local Utilian. Man, those Spaniards need to go. So they use Spaniards as a derogatory term. Hondurans are not, they're not closely related to Spanish people, and they often use it with really explicit epithets like those effing Spaniards. It's really intense. But this rhetoric is also very stressful. Man, those Spaniards need to go. They come here, they don't work, they just steal from us, they rape tourists. And I mean, people will just say this, like they're very open about it. And this is actually from the Bay Island newspaper, which is the main news source for all of the 3 Bay Islands. Camponado is the underbelly of the island. And so after working here for a while, I started to realize that there's a lot of parallels in these populations as what I was seeing in Detroit, where you have a lot of heavy and overt racism and discrimination, a lot of potential for stress, and you also see a huge bias in metabolic disease risk, right? And so this is just some work looking at breaking down metabolic risk factors. In individuals over 40, Hondurans, for almost all categories, have significantly higher risk of these different metabolic risk factors. And the kind of most starkest contrast is with Honduran women, who are the highest risk on the island. Almost 70% of Honduran women have 3 or more metabolic risk factors, which means they have metabolic syndrome. And metabolic syndrome is a huge precursor to any of those actual metabolic diseases, so things like diabetes or atherosclerosis, etc. So this is why I focus specifically on Honduran women for this project. This is just a little bit about the women in the study. There's 134 women. Their mean age was 39. Mean BMI was a little over 29, which is already quite overweight, actually. Median income is about $350 a month. And education was about 8th grade education. And this is, I'm not going to go too much into the data collection, but I'm happy to talk to anybody about this later. Trying to get at these questions requires you to collect a lot of different kinds of data. So I have a lot of really dense interview data, health history data, and then also collect a lot of biomarkers. So for instance, in the study, just for the 134 women, I analyzed over 1,300 saliva samples to look at diurnal cortisol, and then about 300 diurnal blood samples to get changes in immune function over the day. Now this is just some descriptive stuff. So each one of these dots is a woman's cortisol level at that point in the day. And so as we would expect, for the most part, they do follow this kind of trajectory where they peak a bit after they wake up, and then it falls pretty steadily for the rest of the day. And this is also, so I think this is super cool, just because this is actually never, so this diurnal immune function stuff has never been done outside of a laboratory, which, as we know, the real world is messy, right? And there's a lot of noise. So I'm happy that this still picked up this signal where you do see a higher level of lymphocytes in the afternoon versus in the morning. And for granulocyte counts, you actually don't see that much of a change, even though that is shown in a lot of lab studies that you do see an increase in granulocytes. And this actually goes, again, I won't go into this, but I'm really interested in ecoimmunology and kind of when would we expect certain environments to, how would we expect immune function to vary based on environment, where you might expect in a high pathogen environment, it probably makes sense to have a pretty chronically upregulated inflammatory or base inflammatory response, right? But then also this is just a check to see whether we get what we expect in this population as far as the relationship between diurnal change in cortisol and diurnal change in immune function. And so we do find that essentially the steeper the decline in cortisol over the day, the steeper the increase in lymphocytes over the day, right? So on the x-axis here, you have the cortisol slope. On the right-hand side, that's 0. That's like having 0. That's having no change in cortisol. It's very blunted. And the farther out to the left you get, you see a lot steeper of a change in cortisol. And this is the percent change in granulocytes over the day. So the more blunted the cortisol, the less of a change in lymphocytes. Yeah? Okay. Now this is, to the first study, I was interested in looking at how social adversity is linked to the regulation or the crosstalk between those two systems. And I'm going to kind of go over briefly again, this glucocorticoid resistance is what we're looking at here. So the question was, does experiencing social adversity influence the crosstalk between your HPA and immune systems? The prediction was that less HPA and immune crosstalk, which is a proxy for glucocorticoid resistance, will happen in individuals that have experienced persistent psychosocial stress versus those that have not. And how I am operationalizing this is, so you should expect a strong inverse association or correlation between change in cortisol and change in lymphocytes in so far as these systems are signaling properly to each other, right? It's as long as you have glucocorticoid sensitivity, right? So you should see that the greater the increase in lymphocytes, the steeper, oh, sorry, I realize it's flipped from how I just showed it, but it's the same relationship. It's an inverse relationship where you have steep cortisol is aligned or is associated with a strong increase in lymphocytes, right? However, what my prediction is that individuals that have experienced persistent or chronic psychosocial stress should have less crosstalk between those systems, which is a signal of glucocorticoid resistance. So I am suggesting that those individuals, you shouldn't see such strong crosstalk between their systems. Does that make sense? Okay. The domains of social adversity that I focused on for this population were discrimination, loneliness, and depression. And that was kind of for two reasons. One, just from talking to the women over the years, I realized that these were quite salient categories. As I said, discrimination is very overt. People also, so it's rampant, actually. Women oftentimes migrate to the island alone or just with their children. And also, I mean, even people that maybe did come to the island with a partner, it's not a stable partnership, really. And you see a lot of single mothers there. Even women that are in relationships, they still might feel quite isolated from their friends and family just because of the kind of the male dominant culture there, or kind of the machismo culture, I should say. And another reason I chose these categories, apart from being salient to the women, thinking about how we would expect over the course, or I should say how we would expect your HPA system to be sensitized to social cues. What were the ones that would have been salient over the course of human history? You might expect across these domains for those to be pretty strong triggers. And so these are just some quotes to kind of get at this again, women feeling this way. So Utilians, they refuse to help us. They think of us as invaders. And then we also say, okay, well, yeah, there's more. So I was asking, do you prefer to live on the island or on the mainland? So they say, or a woman has said something like, okay, so there's a lot less delinquency here, but I prefer the coast because my family lives on the coast. All right, so now operationally, the way I asked these questions, for depression, I said, how often do you feel depressed? And women could answer, rarely, only on occasion, sometimes, a lot of the time, or almost all, or all of the time. For individuals that answered either sometimes or more, I asked a follow-up question. I said, is it ever so bad that it affects your daily activities? And this is actually what I used for depression because I was trying to suss out kind of this more chronic or persistent depression that's having a real strong impact versus sometimes, you don't feel good for a day or something, but you kind of rebound from it. For discrimination, I asked, in the last year, has anyone tried to make you feel inferior for any of the following? And they could have answered across a number of domains: ethnicity, sex or gender, religion, style of life, etc. For loneliness, how often do you feel alone, or do you feel like you're lacking company? They could answer rarely or never, sometimes, or most or all of the time. So now to look at how these broke down, this is just, this is across all, but then remember I asked that follow-up question of these women that answered more than just rarely. And now this is actually out of everyone though, 41.7% of women experienced depression that it was severe enough to affect their daily activities. For discrimination, this is how it breaks down over the different categories. And 27.5% of women reported having experienced some kind of discrimination, so at least in one of those categories. And then for loneliness, 57% of women experienced somewhat frequent loneliness. Now, to get to this question, do individuals experiencing persistent social adversity have evidence of glucocorticoid resistance? This is for individuals that were depressed, and I'm going to walk you through this. So on the x-axis here, you have percent change in lymphocytes. And on the y-axis, you have diurnal cortisol slope. So what we would expect, again, if there's good signaling between those systems, that we would see the strong inverse relationship between cortisol and diurnal change in lymphocytes. We do see that for people that are not depressed, right? So we see what we would expect. However, for people that have experienced, or that are chronically depressed, we see no crosstalk almost between those systems. And so I do find evidence of glucocorticoid resistance for people that are depressed. Now, what about people that experience discrimination? It's a very similar thing. So that this purple here, those are people that have not experienced discrimination over any of those domains. And we do see that really sharp inverse association between change in lymphocytes and change in cortisol. But for people that have experienced discrimination, we see almost no relationship between the crosstalk of those two systems. And then finally, for people that have experienced loneliness, so this red is people that have not experienced loneliness, and the black are people that have. So there's a bit of a trend there, but it's not indistinguishable. So I'm going to say no to that one. Now I'm going to go through the second study, and then I'm going to wrap it all up and give you the summary of both together. So the reason I'm doing that is because this is also, to my knowledge, the first study that has followed this pathway within the same individual. So we're actually going from social adversity to this pathway of how these systems are regulating each other to an actual metabolic outcome. So now we're just focusing on the second part of that. And I asked, does the crosstalk between diurnal cortisol and changes in lymphocytes relate to risk for diabetes or high fasting glucose? And the prediction here is that individuals with evidence of glucocorticoid resistance will have higher fasting glucose levels. What this is going to look like, so again, you have on the x-axis, you have the diurnal change in lymphocytes, and on the y-axis, you have the diurnal cortisol. And so you would expect strong crosstalk between these systems if you have glucocorticoid sensitivity. And what I actually did is I plotted individuals that had not experienced discrimination, depression, or loneliness. And I plotted the crosstalk, so essentially how these two factors were related in all those individuals, right? And then I extracted a population level mean, and that's what I'm calling the optimal crosstalk between those systems. I then plotted everybody against that optimal slope. And I extracted the residuals. And so, for instance, this person, they're pretty close to that optimal crosstalk. So they're going to have a residual that's pretty close to 0. Whereas this individual, the crosstalk between their two systems, falls quite a bit off from what that optimal level of crosstalk is. Does that make sense to everyone? Okay. And then, so then I use these residuals in the next model to predict glucose. So do individuals with glucocorticoid resistance have higher glucose levels? Well, I found that yes, they do. And on the x-axis here now, this is where you have the residuals from that model, so the crosstalk between your cortisol and your lymphocytes. And on the y-axis, I have fasting glucose. And so on the right-hand side is 0, so that would be being on that optimal line of crosstalk. So the farther to the left you get, the more one is away from that, or the worse the crosstalk between the systems is. And it does track with fasting glucose. And this is significant even if you take out these outliers. So in summary, we see that changes in diurnal cortisol are linked to complementary changes in diurnal immune function. Women that experience depression or discrimination do show more evidence for glucocorticoid resistance. And glucocorticoid resistance is linked to higher glucose. So the takeaway here is a breakdown in the stress and immune signaling system appears to be a pathway through which chronic social stress can influence the risk for impaired glucose. Now to kind of bring it back together, or to tie it back up, why don't we see metabolic diseases across all populations then, right? Is there something about this kind of contemporary context that's leading to us seeing specifically high risk in certain populations and not others? And I would say that perhaps not that inequality is a novel thing in evolutionary history, but perhaps the potential for such chronic stress over inequality, and the fact that there's a lot of domains in which we can have inequality and potential things that might potentially stimulate stress or chronic stress, that might increase the risk. And this is actually, I did do some work where I actually used those different categories of discrimination, of potential stressors. I actually put those in a model together, and you actually do see that the individuals that experience stress over more domains have worse crosstalk between those systems. So essentially every additional domain over which one experiences stress, it is linked to worse crosstalk between those systems. And does it have something to do with our social network or our kin groups, right? We live in quite extreme social isolation, and so there's been a lot of awesome literature that's looked at social buffering, or the ways that we can buffer stress, and perhaps that as we get more isolated, that we are not able to buffer the effects of these experiences of stress. And then I think that a massive elephant in the room obviously has to do with dietary factors and a social gradient in health. So I had mentioned earlier that these systems, the regulatory systems of your HPA axis and your immune system, they evolved under relative nutrient constraint, right? And so they probably function very optimally in that context. But you then put these optimally regulating systems in a context where you have loads of calories. And in particular, there's a social gradient in health, right, where individuals that are lower SES tend to also be relying on more high carbohydrate, low nutrient foods. So can these things then exacerbate or lead this kind of optimal crosstalk or this system to having kind of maladaptive outcomes? And these are some of the things that I've been working on at ASU. And in addition to that, I'm actually, I was talking to Molly about this for a minute earlier. I am working with some really awesome folks at ASU. So this is Melissa Wilson-Sayers and her postdoc Haney. And these are my two mentors, Ken Buteau and Ben Trumbull. I was really ambitious for my dissertation. And I also collected diurnal RNA data. And so I'm just now, as a postdoc, delving into this, where we can actually look at diurnal expression of inflammatory markers in RNA-seq and get a little bit deeper in these questions. So do we see the effects of chronic stress on diurnal expression of inflammatory pathway genes? And also, I have all this data on people's parasite exposure and things like IgE, which is a marker of past parasite exposure. So we can also ask questions like, does parasitic infection moderate how social and ecological stressors influence signaling between those systems? There's been some really cool work by Rook and Raison and some others that have suggested that one of the reasons that we might see both an increase in metabolic diseases and psychiatric pathologies in urban areas has to do with this kind of loss of co-evolved parasites, so that your immune system no longer has anything else to worry about, so it's just overreacting in a huge way to even social stressors as well. And then further, I also collected parallel data between Honduras, and I work with two populations in Bolivia, the Chimane and the Mosa-10, so I also have RNA-seq data and commensurate these social interviews and parasite data, et cetera, between these populations. And so these are populations that are at different stages of the nutritional and epidemiological transition. So we're trying to get a bit of traction on kind of what there's a lot of things that change in that landscape between being going from being subsistence-based and having like you see no metabolic disease in the Chimane, to being in Utila, where you see huge amounts of metabolic disease and they're totally non-subsistence-based anymore, right? So can we look at some of the aspects of the changing environment and kind of parse out what might be the most salient factors and how they interact? And also, I think this is I'm super stoked about this because we're just starting this too, is do the pathways to disease phenotypes vary based on ecological variation? This gets into that ecoimmunology perspective of, do we see, for instance, the relationship between depression and diabetes risk look the same? Are there canonical features to these genetic pathways that might link these certain features to disease risk, or are they variant dependent upon, again, what ecology that immune system, et cetera, is in? And again, what are the role of parasites? And then finally, this is just some kind of way next steps. So can we use the framework presented here to make predictions about how chronic stress influences a range of diseases? If I'm thinking that a lot of this disease risk is actually in the dysregulation of the diurnal relationship between your stress and immune system, that then should be able to transport in different ways to atherosclerosis or cognitive decline, et cetera. I won't go too much into this because I'm already over time, but this is just a longitudinal project that I'm kicking off across these populations to look at the canalization of these traits. And then getting more into this idea of coping and resilience. What are the social and ecological factors that can buffer against disease risk? And so with that, I will say thank you guys, and thanks to my great mentors from my PhD and the awesome people in Utila. And thanks, and I'm happy to answer any questions.
for a great talk. Really interesting stuff and interesting data. I was wondering whether you could talk about the degree to which you saw blunting in the lymphocyte diurnal patterns, because it looked, at least from my understanding of the results that you presented, that the breakdown in that crosstalk seemed to be mostly attributable to court blunting, like court diurnal blunting, and all of the residuals were actually negative.
So
all the dysfunction. You know.
The residuals weren't all negative.
Oh.
I just, because my, I can show you, my hypothesis was specific, or my prediction was specifically targeted towards the like having a breakdown crosstalk, not like very good, you know, like better than optimal, because I'm not sure if that would be better than optimal crosstalk. But this is actually, so I just constrained that figure to show this is what happens if you extend it out. And so here, you know, all around here, there's not too much of a signal. And here, this, even though it goes up, this isn't a significant difference. So that doesn't track to glucose at all. And so there is, that's a really interesting question, because there's two things I think that can happen, right? You have, we do see that the blunted, I mean, going back to that other figure where you just are looking at the relationship between diurnal cortisol and diurnal lymphocytes. So you do see that for some individuals they have blunting in both systems, and yet you're still seeing crosstalk between those systems, right? And I think that one of the things that I'm interested in actually applying that to is thinking about not necessarily, I don't see why that would influence glucose so much, because glucose is undermined by glucocorticoid resistance, but I can imagine things like atherosclerosis, because essentially what happens is this tracks, sorry, not this, let me actually go back to this. This diurnal, here, this tracks lymphocytes, but not granulocytes. And so what you have is that in the evening, you have both a greater, when you have blunted cortisol and a blunted change in lymphocytes, you still have the same level of granulocytes. And so what that means, though, is that you still have high inflammation, but you have a suppression of the components of the immune system that are doing a repair from the damage of the inflammation. And so I can imagine that this might track with arterial damage or something like that, where you're having hyperinflammation, but you don't have the repair. Because this is something I've thought about a lot too, because there are some populations where you have high inflammation, like the Chimane have a lot that have really high inflammation all the time, but you don't see atherosclerosis, and so why? And my thought is that maybe the whole system is upregulated, where you have high inflammation, but there's still a strong diurnal signal in that inflammation, so you still have the repair, where if you have a suppression in the ability of these lymphocytes to repair, do you know what I mean? Like, so then you have excessive damage. I'm super interested in that, though, which goes back to, like, can we use this framework to look at other diseases and make predictions? Thanks.
So it's really interesting material. I realize that you're sitting on a mountain of data and you have a lot already with the RNA material, but it strikes me that maybe a future direction that you might want to entertain would be the causes of individual variation in susceptibility versus resilience. So if we look at your independent variables, depression, discrimination, and loneliness, those are all subjective, right? So by your ethnographic sketch of the social context, it sounds like, well, the word you used was rampant, right? The discrimination is everywhere, but only around a quarter of your participants are reporting that they experience discrimination, right? So there are multiple possibilities. Maybe people kind of live in different worlds, that people who don't interact with the Utilians as much. But another possibility is that there's just individual variation in how much of that sinks in versus it just bounces out. I completely agree. Depression, clearly that's the case, right? Two people can have exactly the same circumstances and one person experiences depression. And the same for loneliness, right? That people can be surrounded by others and feel lonely, or be physically alone and not feel lonely at all, right? So the direction of causality is not entirely clear clear in the work that you have so far, and that greater reactivity. I mean, it's possible that's just a psychological dimension, the susceptibility versus resilience, but it's possible that psychological dimension itself is reflecting differences in physiological reactivity, that there are different strategies, as it were, right?
I completely agree. Yeah, and I would love, one of the things I wanted to kind of bring to the group here over today is trying to figure out how to operationalize or think about how to evaluate differences in vulnerability and resilience. And I do have actually data that I didn't present it here because it's still like very much in the kind of preliminary stages. But so people that said that they experienced discrimination, I had follow-up, like, how did that make you feel? You know, and some people said, oh man, I felt horrible about it. It made me so sad. Other people are like, I don't care. It doesn't bother me that much. And even with, I also have questions about like how much people engaged with tourists. Do they work with tourists? How much their income relies on tourists? How do you consider them your friends? And because you would expect also, some of my earlier stuff was looking at kind of the relative perception of one's socioeconomic status, right? And it kind of gets into that in different domains apart from just socioeconomic status. But if you work with Utilians and tourists, and they become your frame of reference of who you want to be liked by, or who you want to mimic as far as socioeconomically or whatever, I imagine if you can't do that, it's going to feel a lot worse than if you don't care. If your reference population are maybe your friends or family on the mainland who you have a lot more than, right? I think that's super interesting, and like again, that gets into the meat of really, I think one of the things we know the least about is what distinguishes different kinds of vulnerabilities between people.
Do you have any life history data on individuals beyond the time of birth, prior to the time of interview?
In what domains? Yes.
So their lives, the level of deprivation that they experienced prior to migration, deaths in the family.
I do have
other children, siblings.
Yeah, I have like a lot of the demographic data. So for the women, I have the history of how many kids they have, how many of them are alive, or live kids versus total number of pregnancies, or versus kids that they hadn't died. I also have like when the length of time on the island. So when did they migrate and where did they migrate from? And then I have also questions that look at they're right now they're really cursory, and I want to get kind of more focused in this about when you were growing up, did you kind of want for any of these things versus now? Do you kind of, not in the same question, but two questions or a couple questions about those? So some, yeah. I really want to get into that data.
That might be an important dimension of individual differences.
Yeah, I agree. Thank you.
I think Nancy Krieger's discrimination scales in the health inequality literature do a little bit of what you were talking about, which is sort of parse apart and then weight discrimination experiences based on the degree of stress that they conferred on the individual, in case that is a useful scale. But I was wondering whether you could speak a little bit more. I'm hoping I can tap into more of your extra slides to what you're talking about, parasite stress, and any relationships you saw between the ability to maintain this homeostatic immune response or immune diurnal pattern with relation to parasite load or parasite stress across the lifespan. I
don't have that in any of my slides yet because I'm still analyzing the I've analyzed the IgE data, but it hasn't been integrated into the data sets yet. Let me try to think, because I did just look at parasite, like current parasitic infection, and there. So I remember, so in models that include it as a moderator of how the diurnal cortisol is related to diurnal changes in immunity, it had an effect, but honestly I can't quite remember if it was significant. I don't want to take too many steps in case I'm wrong, so I can't remember. And
it's interesting because you mentioned this idea of obviously this Rook concept that parasite load is itself regulatory or imposes some sort of homeostatic regulatory function.
Yeah.
And there's some parallel very interesting that you brought up this relationship between these two systems, because it sort of looks a little bit like social deprivation and court response in early life development in these critical periods where, you know, like Romanian orphanage kind of, you know, like Megan Gunner studies where diurnal court is blunted in instances of social deprivation, where you might have something more like parasitic or, you know, that kind of deprivation conferring a similar effect on the immune system, where you have these like early life critical windows of deprivation. And
yeah, I think that's going to be, that is a really interesting question. And I mean, I think it can go a couple ways in that. So this is just borrowing from the Rook and Raison literature again, but this idea that these systems do better regulate each other so this is actually why I want to incorporate the IgE data, because it's not necessarily that if I introduce parasites, if I give you parasites right now, all of a sudden your immune and your cortisol system are going to start like talking, you know, really precisely to each other. It's more that if you have a history of that exposure, have you essentially, has your immune system been trained to be less reactive to the effects? We can call them social effects, but it might also just be the influences of glucocorticoids, right? Yeah, that I'm super interested in seeing what that would look like, because you would. I mean, the thing is too with like the studies that have found both psychiatric pathologies and metabolic pathologies that come up a lot more in urban, like less pathogen-heavy environments. And you might be able to get some traction on really what's causing that, or what's not. Again, it's not that it's one thing, but this exacerbatory effect, right?
How does your data compare to data on other disadvantaged populations, say minorities in the United States, for instance?
Sorry, say that again. About
how your data compares to data of.
That's a good question. Well, so there have been a lot of studies that have shown that across a lot of dimensions of social, low social status or discrimination, that those things have tracked pretty closely to this blunted cortisol. And also, again, if you look at on the absolute level, again, it's been linked to high and low cortisol. And there's also been some interesting studies. Actually, Steve Cole, who is here, did some cool research looking at across similar domains that it did influence the suppression of different types of immunity and things. So we see similar stuff, but it's hard to speak about the diurnal, like how these diurnal systems talk to each other, and I don't want to like, I'm quite sure I've never come across any studies that have looked at that. So I think that it'd be cool if there were studies that were done in the US-based populations to see. And getting back to the parasite thing too, would we expect this actually to be worse even in US-based populations where there's low parasite loads? I think it's a really interesting question. Yeah.
I'll ask another one. So with these kinds of areas, I think it's very cool that you're looking at diurnal patterns, and I guess I'm aware that they're sort of two like time constructs that people often look at, which is diurnal pattern and reactivity, as like two separate kind of elements of the system that can become dysregulated. Or like, for instance, in cortisol, you might have a blunted diurnal cortisol pattern, but you also might have blunted reactivity to a stress or something, and you would have a similar issue happening in immunology. And I don't know if you have any like speculative ideas about whether there's any adaptive function to dysregulation in either aspect. So like dysregulation, or what we're calling dysregulation, whether blunting itself is functional. It's a kind of odd dysfunction to witness.
Well, but this is the whole reason that I want to do this kind of a more longitudinal or intergenerational study to look at these things, because one could make the argument that if you're blunted, and so what appears to be low reactivity is actually you're just meeting the threshold of what you need to meet, but you just start higher, and so you don't have to jump as far to hit that, right? That could be not always the worst thing, but also thinking about the other factors that can buffer those physiological processes. If for instance the HPA system is blunted in one generation and then that becomes canalized in the next generation, right? So then kids are just starting, which we've seen some of that, at least preliminarily with epigenetic work, right, where it looks like so that blunting can transfer over. That I don't want to use the words like adaptive or maladaptive, but I can imagine then if that is your baseline start, then your body is probably will find ways to not ameliorate, but to respond to that new norm, right? So it might not just be it might not be negative, it just might be that there's a shift in what your body's response look like in the context of experiencing stressors or immunostressors. Yeah, that's again super interesting. Even looking at like the changes in glucocorticoid sensitivity, which has also been a thing that can change intergenerationally, right? It's awesome.
I'm just repeating what you said earlier to add to that conversation, which is that the chronic stress probably isn't evolutionarily novel. Perhaps the degree of inequality increases the level of stress, but then again, ancestral populations were probably exposed to famine and violence in ways that, say, contemporary urban populations are not as deprived and as violent as those neighborhoods can be. But it's the added fuel to the fire of the dietary changes that are potentially really detrimental. So you can expect a trade-off where, if you get blunting because that's a response to chronic stress, that was trading off longevity for immediate resources in dealing with an emergency, and there was a cost to that in EEA, but the cost of that is greatly elevated in a world of caloric superabundance because the times in which in the ancestral environments you experience those kinds of stresses would have been inversely correlated with caloric abundance, and now they are not.
And there's some interesting work too in the animal literature that's even looked at insulin resistance. We think like insulin resistance is bad, but it's not. It can actually be quite adaptive. If you think about like bears or animals that hibernate, putting yourself into a state of having insulin resistance can actually be quite beneficial because it can help buffer against times of not having resources. But I think that exactly this, when you see the negative manifestation of that is when you have essentially an environment that's constantly overrun with too many calories, and I think that's kind of like always the elephant in the room with this research, but it's very hard to get dietary data, or really reliable dietary data, but there was just a study out a day or two ago, I think, that was also kind of looking at these, like comparing different diets, and they're like, no, it's actually the fibrous diet that is the best one, and it doesn't really have that much to do with this, carbohydrates are not the devil, neither is high fat. It's yeah, right, because there's populations that have high carbohydrate, but again, it's like how much is high? It's not. It's an overabundance of calories in general. Yeah.
On that note, that's when you alluded to it as the aspects of diet and BMI, which would be expected to co-vary with elements of diet. If you were to try to bring these results back into the picture of the full suite of ways in which psychosocial stress can influence risk factors for disease and incorporate the element of dietary decision-making, or just to imagine, like, if we could think about, how much of the psychosocial stress effects are you capturing when you're controlling for BMI, for example, versus if you had I'm not in any way trying to criticize the framework you've created.
No,
here.
it's a great
This
point.
is really an elaborate system, and it takes a lot of those controls to figure out how it operates. But if you were to think about how these elements of psychosocial stress may predict
Right.
Or some of the things predicting psychosocial stress, like
Right, does
income
BMI
and
predict, or
such.
does psychosocial stress
Yeah.
predict BMI?
Factoring into the
That's a great
dietary
question.
decision making. How much of a change in disease would be expected if you were narrowly looking at through this landscape of immune cortisol reactions relative to psychosocial stress, whereas if you were to take into consideration also how psychosocial stress influences dietary choice, physical activity, and those downstream influences on.
That's a great question. And I'll say, for instance, in the specific models I ran, it doesn't actually, BMI is significant. It is a significant predictor of, I have more slides. Hold on. It predicts inflammation, but it also does so with, I don't have it. I was thinking that this is what I was trying to show you, but that's not true. Just looking at depressed versus non-depressed and their glucose levels overall. That's not what I want to show you though. You do see that. So you do see that people that are depressed, for instance, I have other things that show that people that are depressed also have higher BMIs. And not, if my memory's not failing me, I don't think that you see that with people that are discriminated or are lonely. But I mean, that there's a psychosocial thing there, right? Where how does your level of depression or whatever influence your dietary patterns or habits? But then also, I mean, one of the things that I want to also analyze is that what is the relationship between the hormones that are underlying your dietary preferences. So there's been some cool study that's looked at increases in the hormones that make you hungrier more, that are upregulated in people that are depressed or feel bad about them. And I feel like we all know this. If I'm super bummed out feeling, I'm not looking for a salad, right? Like, I want some French fries. And so, like these things are all really entangled. The reason I had originally controlled for BMI is because we really don't know the causative directions between like BMI and cortisol. We don't know if having high cortisol leads to greater fat deposition, or if there's something about having high BMI that leads to messed up cortisol. I think that it's more towards having dysregulated or blunted cortisol leads to a better or a greater deposition of fat. But yeah, it's still a massive web. That would be cool to actually look at that, to some of the subset stuff that I have with the Chimane and mapping it onto the really good, because they do have really good dietary data and stuff we were talking about earlier.
I don't know too much about this, but I was just wondering, is there a reason you're measuring fasting glucose versus hemoglobin A1C, which I think is like longer term? Yeah, no, HPA-1C is definitely a much more gold standard. Honestly, it was because when I was down here doing the research, I was using a lipid panel that did total cholesterol, LDL, HDL, triglycerides, and glucose. And so that was the way I was capturing that. But if I were to go back and do this again, and actually with the Chimane and the Mosetan, I've been using HPA-1C because it is more stable. Yeah, that's a great one.
So some of this cohort you're looking at were immigrants, I guess. And so is there any effect that you're interested in relation to the fact that they've actually just changed location? Like anything about not just the fact they're discriminated against or that they may be an ethnic minority, but like actually the fact that they have relocated in itself that's meaningful? Yeah, and that's, this speaks to what Dan was asking about earlier. As far as kind of the data that I have, because definitely one of the longer term goals is to think about trying to parse out one's motivation for migrating and how that would influence their perception of their current environment, and also where their network lies still. That's a big thing still for me, is like, are they mainly tied to the mainland and they feel, you could imagine that being beneficial if you think about perceptions, if their
perceptions of relative success or whatever it would be is more oriented towards where they came from, because they're probably coming from an area that has a lot more struggle than the island. I mean, they're very discriminated on the island of Utila, but the mainland Honduras is, a lot of people are fleeing because it's dangerous and because they don't have any opportunities. And so it could be that they're in a better place now versus if they've shifted that, what their perceived ideal social network is. I think that those two groups of people are going to have different responses to their environment. Yeah, I think that's definitely a future call.
Thank you. So chronotype is like an individual difference variable, right? And so people vary in whether they're morning people or evening people. And as I understand it, it's not just a matter of like when a person prefers to be active, but it is also related to the patterning of cortisol. And I was going to ask you, is that also true about immune function? Because you have this adaptive story of how, well, it's better to have immune function upregulated when you're sleeping anyway.
Right.
But I was wondering, is that true of everyone, or is there individual differences?
That's a great question. I don't know. I wonder if there's been literature that's looked at whether chronotype matters, or what its influence is on the diurnal immune system. I haven't ever seen anything like that. That's super interesting, and right, because that also makes you think. Thinking about what we call, as I don't tend to like to use the word dysregulated. I know I used it a bit here just because I think people are like, we know what this means, dysregulated. But dysregulated seems to mean like there's a pathology there, right? And so it could just be a difference in what an individual's natural rhythm is. That's a great question. Thanks.
Chronotype usually doesn't vary too much outside of Western populations as it does here. Probably because of electric lights, insulation, things like that. They're very constrained in how much their chronotype can vary from person to person.
Oh, cool.
So that's why we don't know the answer.
That's super interesting.
More questions? Okay, thank you.
Thank you, guys.